Um, uh, Welcome to the University of Toronto Department of Surgery, Ben Alman Research and Science Rounds. A Happy New Year to everyone. As you've heard, the discussions that are on our minds these days, of course, are related to COVID, but there's a lot that's occurring that is not COVID related. I wanted to acknowledge a few people before I go on to introduce Professor Scherer, our distinguished speaker this morning. So I want to recognize our chair, Professor Jim Rutka, and thank you, Jim, and the department for sponsoring these rounds, which are, of course, are now being held virtually, which does represent a shift in our practice, and perhaps over the next year we'll gradually start seeing a transition. And I also want to recognize Professor Ben Alman, for whom these rounds are named. And Ben is a past vice chair of research in the Department of Surgery, and now is chair of orthopedics at Duke. Ben, I don't know if you wanted to say a couple of words quickly.
Yeah, so listen, again, thanks everyone for putting together these rounds, and thanks a lot for Steve for coming to speak. I'm really sorry, I usually try to cut up to Toronto to at least see all of you in person. So at least it's nice to see all of you virtually. I hope everyone has a great new year, and I'm looking forward to in 2021 for sure being able to come there in person for this.
Well, it's great to have you here, Ben, even if it's virtually. So. We have over 400 faculty in the Department of Surgery, and research is an important part of the mission for the department, which is ranked among the top 3 in the world internationally. And we're entering into a different era in medicine, and I think in surgical practice as well. And this is the idea of personalized medicine, and the idea of kind of looking at genetic variations in terms of the phenotype and the presentation and the response to surgical treatment. And this was really the inspiration for me to invite Professor Scherer this morning to give his presentation. So just a few words on Stephen. Steve and I actually came on faculty around the same time, and I remember, Stephen, that you and I shared a photo op. We received a Premier's Research Award, and I can remember having a very nice evening, and I was learning a little bit about your work at the time, and I've been following your work very actively. We both were part of an NCE in neurodevelopmental disorders for a number of years. So Stephen holds the GlaxoSmithKline endowed chair at the Hospital for Sick Children. He is director of the University of Toronto McLaughlin Center, as well as the Center for Applied Genomics at Sick Kids. And Professor Scherer's landmark discovery really relates to the discovery of global gene copy number variation as an important and common form of genetic variation in human DNA. And this discovery really launched seminal investigations in a number of disorders, but particularly related to autism spectrum disorders. And his work has been very highly cited. Remarkably, his H-index is well over 130, which is really in the stratosphere. And he's received a number of awards. I'll just mention a few of these: Howard Hughes Medical Institute Scholarship, Killam Prize, a number of honorary degrees, and he's a fellow of a number of societies. But I'll point out that he's a fellow of the Royal Society of Canada, and his work has been singled out by Thomson Reuters as being particularly noteworthy. And we could have invited anyone in the world, particularly with the use of Zoom technology, but I think the most qualified person in the world, quite frankly, is Steve Scherer, to talk to us today about his work. And his lecture is entitled Gene Dosage Variation in Autism Spectrum and Related Neurodevelopmental Disorders. And Steve, thank you very, very much for sharing your amazing research with us. And I hope that we'll be able to think about ways to apply this in our surgical research and our surgical practices. So I will stop sharing my screen, and assuming the Zoom technology works, Steve, if you can share your screen. Thank you.
Okay, how does that look? Good to go? Okay, thank you so much, Michael, Dr. Failings, Dr. Rutka, and Dr. Alman. I've known Ben since I think it's 1997 or so. So it's really a great honor to be invited to give this presentation this morning. As Michael said, the title is Gene Dosage Variation in Autism and Related Disorders. So these are the three objectives of my talk. The first is to update surgical practitioners and researchers on the rapidly evolving understanding of genes and genetic variation, in particular gene copy number variation in the human genome, including the relevance of this information to the study of human disease. That's really the basis of my research for the last 20 or so years. Second, to discuss how the new genomic technologies, in particular whole genome sequencing, has helped to define gene dosage variation. And what that means is having one copy of a gene instead of the typical two. And how can this can lead to complex neurobehavioral conditions such as autism and related disorders. And third, to contemplate how genome sequencing technology often moves faster than our ability to fully comprehend the data it generates, and how this conundrum may influence the uptake of new genetics and precision child health in the broader society. And I'll try to seed some comments in of how this is really relevant to the Department of Surgery. But as Michael Failings pointed out in our rehearsal a few weeks ago, really what we're trying to do is use genetics to help subcategorize all the different types of disease. There's over 6,000 cataloged genetic diseases. And as I'll argue throughout my presentation, we think that probably every disease has some contribution of genetics in some way. Even behavioral disorders, which were thought, even a decade ago, really not to have a biological basis, most of them anyways. And this is what I'll show you today, that in fact, for some of these, we do have biological factors identified. So in fact, when I was a graduate student in the late 1980s and early 1990s with Lapchi Choi, of course, the famous cystic fibrosis discoverer, this was my PhD thesis question, really, was what are the number of new mutations that each of us have in our genome compared to those carried by our parents, so-called de novo mutations or spontaneous mutations. And it took, I got my PhD, but it took about 20 years to actually get the answer to this. So we now have this. This is really amazing because we can sequence genomes in their entirety, as I'll show you. And here's the answer. So this is a pedigree, mom and dad, and the F1. And we're really quite similar to our parents. We only have 73 new events in our constitutional genome. These are the chromosomes that we inherit. And these are broken down into approximately 60 single nucleotide variants, so single letter changes, and about 13 insertion deletions. So insertion deletion of larger segments. I'll show you what this is in a minute, or a larger copy number variant. And then roughly 3 somatic exonic mutations that are present in over 80% of cells, so early somatic mutations in each genome. So, when a patient comes into your clinic, the first thing you ask them really is what's your family history? And you can get a lot of data just by asking these questions. And in fact, this is one thing that I learned from one of your most famous alumni, Bob Salter, Professor Salter. In the early days, I think it was just before he was involved in recruiting Ben to Sick Kids, we spent a lot of time together, and he taught me that the best way to know your patient is to know all of your patient, not just why they're coming to see you in clinic. So holistic medicine in a way, not a great term, and this really was foundational for my thinking about how to study genomics, how to study the genome. You need to study all of the genome if you want to understand the genetics. Okay, so the point here is if you can study these new mutations in individuals that have a phenotype, a clinical presentation that's not present in their parents, it's quite likely that new mutation actually pinpoints the gene that's involved in that individual. So that's been the foundation for a lot of our studies, and I'll come to this in a second. To make sense of all this data, you need to have information maps or to compare your data against. And I think probably most of us, at least on my screen here, of the era where we learned in high school and university, the karyotype, that's still the #1 genetic test performed at sick kids, believe it or not, as it developed in the 1950s. You look under the microscope and you see the morphology of the 23 pairs of chromosomes. That's the genome, but in a karyotypic form. So this is the lowest resolution scan. And then the highest resolution scan is to sequence the DNA. That's what you're seeing on the right here, to look for these single nucleotide or single letter changes. And we use this now, the technologies used are sequencing, in some cases microarrays. And then to capture this in-between size genetic variation, I'll tell you what it is in a minute, copy number variation, you can use microarrays or whole exome sequencing. And as I'll show you, this was really what our contribution to this catalog of human genetic variation was. It was the initial discovery of CNV, or copy number variation, and then to catalog variants. So we had a data set, an information resource to compare all of our disease studies against. Okay, now I know a few of you on the line are kind of students of history also, in addition to surgery. The discovery of these genetic variations is really quite interesting. If you think about looking down the microscope to discover, chromosome number in humans, and then, for example, chromosome aberrations, say 3 chromosome 21s in Down syndrome, you're looking at a very low resolution. And then you go to the order of discovery. This was the 50s, and then into the 1970s and 80s, you could do sequencing at the highest resolution. And then the middle resolution of variation was kind of forgotten about because people just didn't know it existed. You didn't have the technologies. And I think it didn't follow the logical progression of size of variation in the discovery process. So it really quite an aberration in discovery of science. So this is where our role was. We had the first microarrays actually in Canada, probably in North America. And we started to look at the genomes using these microarrays. And at the time in pre-2004, if you plotted on the x-axis the size of genetic variants, so on the left is a single nucleotide, one nucleotide variations, there's roughly 3.2 million SNPs in each of our genomes. And then if you look on the right-hand side for these large genetic aberrations, about 0.6% of the population has a chromosome abnormality that would be seen under the microscope. Okay, so then we started to look using microarrays at control genomes and also genomes from people who had autism. This is what I had funding for at the time. And sure enough, as you would expect, because systems always favor equilibrium, all of these other classes of variation were identified. We kind of knew, we should have known that they were there, but we didn't have the technology to find them. So in fact, to cut a long story short, we now know that there's more than a 10-fold number of nucleotides affected by copy number variation than SNP variation in the human genome. So the primary message from the Human Genome Sequencing Project in 2001 is that we're 99.9% identical. We really only had these 3.2 million SNPs, but in fact, that was an underestimate. As I'll show you, there's something like 30 to 40 megabases, million base pairs of nucleotide variation captured by these copy number variants. So what this means practically is genes don't always come in pairs. Like we tend to teach ourselves, this needs to make it into textbooks. Quite often you only have one copy of a gene, perhaps 3 copies, or segments of genes are missing or swapped. So this is what copy number variation, or CNV, imbalance is. Just to show you a little chromosome segment, you can have a reference genome, and this segment in brown and blue may be missing and deleted. Or you could have a duplication, and the duplication could be tandem, a segment just copied over beside each other, which we often see in muscular dystrophy, for example. The duplication can also be non-contiguous, so it could be inserted elsewhere in the genome. Okay, it could be on the same chromosome or on a different chromosome. And then as shown in category D here, you can have higher order replication. You get multi-copy CNVs, and you see this often in gene families. And then there's also these complex forms where you can have inverted segments of DNA, duplications, deletions, and they can be scattered all over the genome. These tend to happen more in cancers or somatic variation, but we see these in constitutional inherited variation often. Once there's less likely pairing happening at meiosis and mitosis because the chromosomes aren't lining up, the replication process gets screwed up even more. And I'll show you one example in autism a little bit later. But the point is, pre-2004, we really didn't have a catalog to compare against. So in most disease studies, we're actually incomplete. And now we know that there can be megabase sizes of these CNVs in genomes of people who can be working front line in grocery stores, for example. I have one example. A megabase deletion, the person is just doing just fine. It really depends on where the deletions happen in the genome, what genes are affected, as I'll show you. Okay, so this is the most important slide in my presentation, and think about this from a lot of different perspectives. You know, when I was in university, we learned, based on what was known at the time, really, biochemistry, that for most proteins that were known, because it was enzymes that were studied really at that time, 15% or 10% of function was enough, right? So, and this is really what these purple proteins are showing here. This is what's called a gene interaction map of the genome. Every single protein coding gene is mapped here on one of these different categories. So the title of the slide is 10% of the genome or proteins are copy number variable. Okay, it's okay that they're copy number variable because there's really no effect. You can have a whole deletion. As long as you have 50% activity, that's enough for an amylase, for example, or some type of an enzyme. And the reason we knew about that is because that's the kind of class of genes and proteins that were studied that were amenable at the time. In fact, the major story in the last 20 years, in my opinion, is that it's this other 90% of the genes that we're now just starting to study. They're copy number stable when you look at controls or population controls, okay? And the reason being is if they don't have two copies, they show up in disease populations. So all of these different proteins and the genes that encode them on the right-hand side here in green will be involved in some disease if it's a deletion or duplicate, something that takes them away from stability or the haploinsufficiency, for example. And I'll talk about these a little bit later. So, you know, if you're going to make a prediction, we probably have associated maybe 10% of this 90% in disease right now. All of these will be involved in some type of disease. The other point is that 75% of the genes that encode proteins in the genome are expressed in the brain. Okay, so the brain surgeons, you have a valid argument that you're the most important form of surgery in the department. Say that tongue in cheek. So, well, you know, the idea really was this, is if we can then have a catalog of all of the copy number variation in control populations, in particular those that don't have an overt or known disease, and then start to catalog other databases where you have CNVs or in particular deletions, for example, in different phenotypes or clinical presentations. You just have to look at the overlap between the two. Okay, and the ones that are on the are never on the left or more often and on the right or more often on the right, using statistical analysis, will pinpoint the genes that are involved in those phenotypes. It's really as simple as that. But because these are rare mutations, remember that you will only see roughly a new CNV in the population in about 1 in 100 individuals. Okay, but the impact is large because it affects so many nucleotides. So you can use this approach by going back and forth. And this is exactly what we did. We started this once we made the observation of CNV. We started already in 2003, 2004. I should say we maintain at SickKids the control database, this database for genomic variants. And we've now cataloged over 2 million CNVs in this database across worldwide population, mainly from samples or individuals of European or Caucasian ancestry. Okay, so we're still way underpowered when we're doing studies of diversity across the human populations, and you'll see more of this going forward. So with many clinical collaborators across Toronto and really around the world, we started to test this hypothesis. If you could just look for these large de novo copy number variants in controls and then in disease populations, You could then pinpoint specific regions of the genome where you have a higher de novo mutation rate hitting specific genes. And this is actually table 1 of a paper we did in cerebral palsy with Michael Chevelle, Mary Mosquet McGill, and Darcy Failings, and other Failings in Toronto here. Say the better half, or I don't know how you want to say it. Michael can comment. And their teams. And really, this is incredible because we had deep clinical phenotype data, and it's so important to have this when you're doing these disease association studies. I'm not a medical doctor, but I learned so much how the importance of the clinical phenotyping is, both pre-study design and post when we go back and do genotype-phenotype correlations. But anyways, we found in the general population, roughly 1% or so would carry a large de novo CNV. And we had a much higher rate in all of these different diseases. I'm going to talk about autism in my presentation, but cerebral palsy, in fact, was one of the higher ones. Intellectual disability, upwards of 20% are de novo CNV mutations. And because of this approach, hundreds of different disease or disorder susceptibility loci have been identified, and I'll talk about a few of them. So the message here is location is important. For some genes, only having 1 copy instead of the typical 2 or 3 copies is a sufficient imbalance. So again, an aberration from what equilibrium is, and that's sufficient to lead to the individual who's carrying it to medical attention. And this was really the basis now. There's over 8,000 clinical microarrays run for diagnostic testing of neurodevelopmental disorders and congenital abnormalities at Sick Kids in Credit Valley in Ontario alone, and well over 1 million run worldwide. This is the first tier genetic test now for these type of disorders. Okay, so I'm moving into the second part of my talk, and I'm going to use autism as our model for the CNV work and also the whole genome sequencing, because we've been doing this for 20 years. It's fascinating. This is a neurobehavioral condition that even 15 years ago, we had no genes in idiopathic forms had been identified at that time. So really there was a question, is this really a biological condition, or is it a construct of interpretation essentially? So what is autism? I'll show you, it's highly genetic. Based on the DSM-4 criteria, we're now into the DSM-5, but I talk about DSM-4 because most of the samples the families were collected under that clinical criteria. But it's a disorder involving 3 deficits in 3 domains of function, extending into the continuum of human behavior. That's what I've tried to draw out here in this Venn diagram. All right, so if you have sufficient impairment or deficits in social communication, language deficits, and restrictive repetitive behaviors that meet the diagnostic criteria, you can fall into Asperger's syndrome, autistic disorder, childhood disintegrative disorder, or PDD-NOS. Now, it's really important that we see in upwards of 50% of cases intellectual disability, anxiety or ADHD in perhaps 50% of cases, and many medical comorbidities. I think this is probably relevant to the surgical background here. But importantly, we see this as a continuum into normal, into human behavior. The features that are involved in autism are those features that actually make us most human. Our ability to socialize, interact, to communicate, and cognition. So in fact, this is a disorder of the human condition, I call it. 1 in 66 Canadians will receive a diagnosis by age 5. And we see this interesting 4-to-1 male to female sex bias, and it goes up to 12-to-1 male to female in, if you consider Asperger's syndrome, for example. Very highly heritability. If you look at identical twin studies compared to dizygotic twins, the heritability is estimated to be in the range of 50% to 90%. So inherited genetic factors are suspected to be involved, but as I'll show you, these de novo events are the ones that led us to the first round of genes to be identified. So the clinical challenge is there's over 100 different disorders that carry different names that also can exhibit ASD. Things like Rett syndrome, Fragile X syndrome, etc. And identical twins that have the same constitutional genome, even though there's a significant genetic component to this, can have divergent ASD clinical severity. roughly 10% or 15% will be on opposite ends of the spectrum, even though you have the same genome. And 20% of parents have what's called a broader autism phenotype. So they don't meet full criteria for ASD, but they have a lot of the characteristics. And we see this at increased rates in alumni of universities that have technical engineering and mathematical schools, for example. So typically it seems to be higher in the fathers. I'll come back to this in a second. The average age of diagnosis in Toronto is about 4.5 years old, but the optimal age of behavioral intervention is much earlier. So getting a diagnosis is really critical early on. And this concept of diagnostic odyssey happens in probably almost every single case, and it's exemplified here by the child on the left who went through, I think it was upwards of 8 different clinics at Sick Kids and across Toronto until he was diagnosed with a chromosome 16 P11.2 CNV deletion in the clinical setting and in our whole genome sequencing study all in the same week. So they really got the diagnosis in 1 week when they were looking for an explanation for probably close to a decade here. Okay, so there's complex queries coming from families. Is it a de novo mutation? What's the penetrance, expressivity? I'll talk all about these as I go forward. I think this is really, from the societal level, quite interesting. Pre-2003, which is the DSM-4, DSM-5 transition, 70% of the families coming into our research studies were complex. So they had classic ASD. And now it's the reverse. It's 70% are higher function, more like Asperger's type conditions. So when we think about, how we perform our research, how we interpret the results, how we deliver the data to families, we have to know what type of family we're delivering that data to and what their expectations are. Okay, so whole genome sequencing. This summarizes 50 years of technology, this slide. And just to point out, so whole genome sequencing, right now we use the Illumina technology. There are other technologies. You can generate short read sequences at a high coverage, covering the 3.2 billion chemical bases or nucleotide bases across the genome. Then we use computational methods to tease out the different classes of genetic variation, which are shown on the left-hand part of the slide here. I mentioned single nucleotide variants, copy number variants. There's these things called trinucleotide repeat expansions you see involved in Huntington's disease and about 30 other disorders. And we had a big paper came out led by Ryan Yuan. I won't have time to talk about this, showing these type of expansions are also involved in autism. It came out in Nature a few months ago. Structural variants are these complex CNVs, mitochondrial variants, and then paralogous sequence variants that are found in things like spinal muscular atrophy, for example, SMN. Okay, so you get all this data if you have the right algorithms and you have the right surgical training, if you will, to actually extract this data. It's very complex. This is what my laboratory does. We have a staff of 30 informatics people who are arguably some of the best in the world who do this. I wanted to point this out, as certainly the people at Sick Kids, but also others across the TAS and hospitals. We'll hear a lot about whole genome sequencing and cancer projects and others. This is good. This is what you want in science. This is what we dreamed about when I was a graduate student, get all of the variants. Otherwise, you're doing incomplete genetics, right? If you're trying to do an association test, if you only have 1/10 of the variants identified, you're going to get the wrong answer most of the time, or an incomplete answer. So you really need to do whole genome sequencing to get all classes of variation. The technical perspective, it's a lot faster than actually doing something like whole exome sequencing because you don't have this intermediate step of pulling down the target DNA. And then most importantly, we only know maybe 1% of what's going on in the genome, right? So there's lots of other discoveries to be made, and to make those, you actually have to have all of the information in front of you. So when we talk about precision child health, precision medicine, whole genome sequencing is the basic currency of information to help you get to subcategorizing the genetic contributors to that disease. In some cases, they'll be absolute. In some cases, there'll be susceptibility or risk factors. So this is my slide. I thought maybe the real surgical techies would enjoy the most. This is the kind of data we look at in the computer in the thousands every single day. We've now developed machine learning approaches to do this, but this is what it looks like when you look at by eye. When you do a whole genome sequence, you can cluster all the sequence reads. Each little stretch here is actually a sequence read of 200 nucleotides or so. and you get roughly 30x coverage, 30 times coverage of a segment of the DNA. This is the gene CHD8, and on a microarray, we would detect this copy number variant. You can see here, the typical 2 copies would be kind of this segment of DNA here. If you have increased reads going up by 33%, which you see here, that means it's a copy number gain or a duplication. When you look at the sequence, we get a much higher resolution of the breakpoint, so it's surgical. We actually see the breakpoints. It's really amazing because the CHD8 gene has lots of exons shown here at the bottom. And we need to know which exons are actually deleted or duplicated to have a proper genotype-phenotype correlation. This one we were able to detect by microarrays, but on the next 2 examples are genes known to be involved in autism. One is a deletion, it goes from 100% to 50% here. And then the second one in C0 is a duplication. Okay, we miss these using every other technology. So you only find these using whole genome sequencing. So our clinical yield when we do the experiments goes much higher. So, you know, my group has invested a lot of time developing this project. We've now collected and followed with clinical scientists across Canada. The names are listed here on this slide, and we're following over 4,000 families across Canada. They're mapped here. These are all people who have now been sequenced using whole genome sequencing. This is roughly a $40 million project we've been doing over the last decade with Autism Speaks and Google. And we are able to map and link in some cases into the health administration databases. So these people are mapped based on their postal code. All of these families have been sequenced now. We have their genotypes. We can cluster them on the top right here. We mapped them across the HapMap control populations. About 70% of our participants are of Caucasian background. These are individuals with Black chromosomes, and then Asians, et cetera. We can map based on genotype. This is essentially what 23andMe does also when you're looking at common genetic variants. We're doing a lot of sequencing of those families that we think are highly genetic because they have a lot of affected kids. These are all pedigrees, and then the black boxes indicate those that have a diagnosis of autism. Okay, I won't go into too much detail here. We actually have one family that has 8 affected kids, believe it or not. No unaffected. And then families where we have, say, more affected females, right? To look for those that might have a bigger genetic load because of this 4-to-1 male to female ratio. And this is one really interesting family now where we've identified at least 13 arms of the family where every single child conceived through a sperm-based donor IVF, a single sperm donor, has some form of ASD. The females tend to be less affected. So we prioritize these and are doing sequencing to try to identify those high impact genetic mutations that appear to be segregating in a dominant mode. And then every single trial, person going through clinical trials at Hull and Bloorview with Evdokia Anaganastu, and then also with some pharma partners are going through whole genome sequencing. So we don't even think anymore, we just sequence now. It's under $1,000. We get so much more data. If we get a DNA sample, my job is to figure out how to pay for it. But we sequence and think about the data afterwards. So we've developed a lot of different ways to look at this, and I won't spend a ton of time on this, but we've taken an open science approach, put all of the data into a massive cloud. This is run with Google Genomics, but in fact, it's actually coordinated out of my laboratory. We have deep phenotype information shown here. You can enter the database in many ways. You can look at the genic level. You can look at the sequence reads. And we actually have a community portal where we can link families who want to link to other families that have the same genetic alteration involved, so they can compare stories. And then we make this open to any approved researcher. There's over 200 approved researchers from 17 countries using this data. The idea was we spent a multi-million dollars to generate the whole genome sequence. We only want to do it once, put it in a cloud, and then let the really smart people with new algorithms take their questions into the cloud to do the analysis actually on our dime. So we have people from all over the world. We have some really smart students in India, for example, or China, or who've got a great idea. They can take their idea to the cloud at our expense and do hypothesis-based research. So this is a summary of really a decade of work. We published it in Nature Neuroscience in 2017. We're going through this whole process again right now with double the amount of data. And I think these numbers will all just essentially double in size. But let me just quickly run through this. We've now identified 61 ASD risk genes that are used in clinical diagnostic settings. So these are essentially appearing as Mendelian disorders, are very highly penetrant. If you carry one copy of these instead of the typical two, you'll have autism or something that looks like it. And copy number variance accounts for 6% of these cases, typically with more severe autism and clinical consequences because the deletions are bigger, they affect more of the gene or genes that are nearby. 30% of syndromic ASD carry mutations, and roughly 5% have 2 or more mutations. This is really important. If you take any genetic disease where you have a genetic diagnosis and do whole genome sequencing, in roughly 5% of those cases, you're going to find an other mutation in another medically relevant gene. So in fact, the patient's going to have 2 or more different genetic diseases. Okay, we see this over, in fact, that's an underestimate. My guess is going to be 7 to 10% once we know everything. So most of your patients actually, not most, sorry, about say 10% of your patients actually have at least 2 different diseases. So it comes back to the Bob Salter approach to, know holistically, genetically. If you want to know holistically, genetically, you need to do whole genome sequencing as early as possible so you know what disease you're actually treating. In autism, 1% are recessive mutations. Somatic mutations are also involved, early occurring somatic mutations in brain development. Now, this is critical. In 50% of the multiplex families, so families that have 2 or more affected kids, the SIBs can carry different ASD mutations because there's so many different genes involved. Okay, and we still have a lot more to be found. We've already identified at least 3 genes, GEFRIN1, astrotactin2, and SHANK1, that are on autosomes, so not sex-linked, that the mutations express only in male carriers as far as we can tell. So there's something linked here to probably hormones. Lots of X-linked genes are involved, many involved in intellectual disability. Okay, so intellectual disability, as I mentioned at the start, and cognition are almost interchangeable in ASD. Lots of epilepsy genes also. About 50% of autistic individuals have some form of seizure. Now, this is unpublished data, but it builds from a study we published in JAM a few years ago, where we looked at combinations of rare mutations. So this 10% or so that have 2 or more mutations or 2 or more genes involved. And it's not surprising, but when you have a more complex form of ASD shown in green, you tend to have a copy number variant mutation involved because it's more impactful, it's bigger, and affects all of the isoforms of a gene and perhaps some nearby genes. And then the smaller mutations tend to be more involved in what we call essential ASD, so those that don't have as many medical complications. This is important for genetic counseling, and it's really from a model building perspective in genotype, phenotype. We want to incorporate all classes of genetic variation to calculate a so-called polygenic risk score. Okay, so all of those rare genetic variants and common genetic variants, so we can get to all of the inherited and the spontaneous forms of autism. We have a long way to go. We probably need something in the order of, I would say 500,000 genomes done, maybe 1 million will be better. We've got roughly 20,000 genomes, whole genome sequencing from autism families right now. Now, this is the second most important slide in my talk. These are the 6 most prevalent CNVs we found in our Canadian cohort. They're all present in less than 1% of the cohort. So these are all rare mutations. Okay, some are spontaneous, some are inherited, but they're high impact. So the chromosome 16p11.2 is the deletion I showed in the child with on the Toronto Star. Orexin 1 deletions, I'll talk about this a little bit later. Duplications of the region that's involved in Williams-Beuren syndrome, that's a deletion syndrome, but if you have a duplication, you have a form of autism. 1q21 duplications, and these synaptic scaffolding genes, 1, 2, and 3. If you have deletions of these, it's very highly predictable for ASD. But the point here is that the specificity is not so high. So you see these same CNVs and schizophrenia, bipolar, ADHD, intellectual disability, etc. And I added even cerebral palsy. I think it may not have made it on this slide, but we see many of these coming up in cerebral palsy, which is a very complex condition. So, I'll talk about this at the very end of my talk about, how we can use this. Of course, we're using it clinically already, but if you're doing sequencing, say, of individuals in the population, how predictive is this? And Ann Bassett has been looking at adult populations and doing very similar studies, and others in Toronto also. And she's found these same CNVs in adults with neuropsychiatric conditions. And when we have data to go back to the kids, either through clinical records, or if it links through families, through say three generation families, and it's all described in these papers, you can actually see that these kids or these people in their earlier years had features of autism. And then in the adult expression of the disorder, you may see more complex neuropsychiatric phenotypes. Okay, so I think the concept of predictive medicine will be very interesting to see what some of these CNVs we're finding more recently are going to look like in these people as they get older. And the best way to kind of capture that is through family studies. We spent a lot of time cataloging this information. Again, we've probably looked at more chromosomes than anyone in the world with respect to neuropsychiatric disorders. And I just wanted to point out this deletion of 1q21. You see it in lots of different conditions. We see ASD in 10% of people who have these deletions, and up to 50% of these are inherited. Whereas the duplication we see is the reciprocal event where you have 3 copies of the gene. We see 36% of people will have ASD. Okay, so 3 copies are more often associated with ASD than 1 copy, and 2 copies is the norm, is the population norm. Also, 2/3 of these are inherited. But this deletion 2q23.1 that encompasses is a very important MBD5 gene, 100% of the people tested have autism. This is quite predictive for autism. And these are all presumed to be de novo in inheritance. These are de novo in origin because these people are severely autistic and they don't develop social relationships and have kids, so they don't pass their chromosomes, right? So this is a concept of inheritance also. So this is very important in genetic counseling perspective because these are de novo mutations. I'll come back to this a little bit later. And then I thought some of you would find this slide quite interesting. I know many of us spent so much time in school. We didn't start our lives to our late 20s, 30s, and maybe even 40s, but age of chromosomes is very important in predisposing to genomic mutations. This is work Ryan Yuen did, but also a group in Decode Genetics in Iceland did similar work. So if you look at these de novo mutations, more often than not, they occur on the paternally inherited chromosome, both for the SNVs, indels, and CNVs. But they tend to be, when they're clustered along chromosomes, and I'll come back to this in a minute, they occur more often on the maternally inherited chromosome. So if you look at the reason why the mutations are more correlated with the paternal chromosome, they actually correlate with the age of conception. In this case, it's actually age of child at birth of the ASD individual. It's the age of the paternal chromosome. So the age that the dads are actually having their kids. It's a higher mutation rate in spermatogenesis, the longer you wait to have your kids. Okay, so when you're over 40, for example, is relatively high, adds about a 3% risk actually to having a child with ASD if you wait much longer. And then if you look at this clustering and mutations along chromosomes, we struggled with this for a long time. Why was it happening? In fact, we see as shown here that if there's a clustering of mutations and there's a copy number variant on a maternal chromosome nearby, you have a higher new mutation rate of SNVs, single nucleotide variants nearby. And we think this has to do probably with the DNA replication process being different in females and males. So the number one risk factors involved in this 1 in 65 prevalence statistic in Canada anyways, of course, are family history, parental age, gender, because of the 4-to-1 male to female. And then the prevalence increase probably largely has to do with the shifting diagnosis. So a lot of individuals have intellectual disability now, for example, get an autism diagnosis. But from the biological perspective, the major contributor is actually the paternal age of the chromosomes. Now, if you take this slide, when we look at those genes, so I mentioned we have 100 genes. Well, what do? They're proteins and code molecules that work in how the neurons connect and communicate with each other. Okay, so this figure is very similar to that one I showed at the start. And many of these genes, actually all of these genes are in those bubbles I showed you at the beginning. I won't go through this, but 80% of the 100 ASD genes are connected into protein networks here that are involved in neuronal development and axon guidance, chromatin modification, transcriptional regulation, and MAPK and other signaling. I know Ben actually works on a lot of these different genes, at least in his previous work. So I'm going to go through this really quickly. This is a kind of a schematic neuron that I drew out of what you would see in a typical neuron of the trillions, billions that you have in your brain. And this is where you have balance of Nerex and SHANK2. So you have two copies. PTCHD1-AS is actually on the X chromosome. Okay, so it's kind of like a tree model, trees and branches and leaves. The Nerexin-1, this is a very common change that we found in our ASD collection. The balance of homeostasis is tipped in the direction of autism here because you have a paucity of branching of the dendrites compared to the typical control. Because Nerexin is actually a structural protein involved in neurons. This is really nice work that James Ellis did here, where he looked at SHANK2 mutations. This is a deletion, so haploinsufficiency, and it leads to hyper branching actually of the dendritic and dendritic spines. Okay, so again, it's just an aberration in the homeostasis. And then I'll show you at the very end of my talk, and I'm getting there, what we see now in this long non-coding RNA, where we see the same morphology, but a change in the excitability of the neuron. So this is also precision medicine where we have the mutation, and then we can see at the neuronal functional level what's actually happening. And then you have to think about which drug treatments might actually modulate this back towards what you see in the typical neuron. Okay, so for roughly 20% of these families, we're finding genetic alterations or mutations that are reported back through genetic counselors as part of our research study. I'm going to go through just very quickly 3 examples. This is a highly penetrant autism gene on the X chromosome. We found the mutation in a male proband, and we're able to actually do the genetic diagnosis pre-symptom presentation, and then this child went through early behavioral intervention and he doesn't have a full diagnosis now, probably because of the intervention. Okay, so he's on the broader autism phenotype. So that's pretty straightforward if you have a penetrant mutation on the X chromosome. It's inherited from the mom. This is one of these multiplex families where we have a de novo mutation in the thyroid hormone receptor alpha gene. So it's not in mom or dad. And we have an inherited mutation in the CATNAL2 gene, which is an epilepsy gene. As I mentioned, in these cases where we have 2 or more affected individuals, in roughly 40 to 50%, they're carrying different penetrant mutations. So it comes back to the fact if you're doing genetic diagnosis and you find a mutation in the older child, you can't just look for that mutation in the second or third or fourth born. You have to do whole genome sequencing to see what's really going on holistically in the genome. So in this proband case, actually, it was the first example in Toronto of a child going on thyroxine treatment, which seems to have a positive impact on his metabolic rate and neurocognition, increased energy. And then this is an example of this gene I'll talk about in a second, which is a 1.7 million base pair loss on the X chromosome. Okay. As I show you, these are typically inherited from mom. It's on the X chromosome. This was the first case where we had a de novo mutation. Really critical because the parents want to know what's the chance of them having a second or third child. Because this is de novo, it returns back to the 1 in 65 of population frequency, right? So it's very important for family planning. And as I'll show you right now, because I got to keep moving, this is really a genetic observation that led us into a whole new genetic pathway. And it's actually a long non-coding RNA. I won't spend any time on this, just for those of you who have been out of school for a while, we're not geneticists. There's as many of these long these RNA-based genes that don't encode proteins as there are protein coding genes. So in my opinion, next to the CNV discovery and this is in the post-genome world in the last 15 years or so. This is huge. All of these molecules, they have function in different ways. 70% are expressed in the brain. Okay, these long non-coding RNAs, we found one that was affected by this deletion I showed you in the last family. It has 15 different variants or isoforms. It spans 1 million base pairs of DNA on chromosome X. And they tend not to be conserved at the sequence level between human and mouse because they don't code proteins, but structurally they're conserved. They occupy the same sites on the chromosome. And in fact, from the clinical perspective, this 1 million base pair region had lots of deletions in clinical microdata, but it was never interpreted because 10 years ago, no one really knew that these genes were important on the chromosome. So people just left to the side. So we were the first really to look at this. We published this first in Nature in 2010. And this is how clinical geneticists look at chromosomes now. And I thought I would show this because this is, this is the new genetics. This is a segment on the X chromosome. This is this long non-coding RNA and exon skipping through this 1 million base pair region. This is a nearby PTCHD1, and I know Ben will know about this protein, 3 exon coding protein involved in sonic hedgehog signaling pathway. These are deletions in patients that have autism, and the extended deletion is shown in red. We have lots of them now. Every single male that we have that has deletion of this region of this long non-coding RNA has a form of autism, and it tends to be high functioning autism, okay? Kind of the essential autism. If they have a deletion of this region here that includes a protein coding gene, they have a more complex form of autism, including intellectual disability. We don't see this in any controls unless they're female carriers passing it to a male affected individual. Just to go quickly through this genotype phenotype slide, it just summarizes what I just said. So the deletions have autism. Typically it's high functioning, communication impairment, including kind of an emphasis in the exon 2, 3, and 4 region of this genome. So we can now take skin cells or blood cells from patients that have these deletions, turn them into induced pluripotent stem cells, and then differentiate those into neurons. And in the Petri plate, we can grow neurons from patients. Again, another form of precision medicine. We have all the sequence. We can translate them into neurons and check the morphology and the branching characteristics that I showed you earlier in that schematic and show that these are exactly the same as typical neurons. So structurally, deletions are the same in these patients. Cut a long story short, then Mike Salter's group looked at the electrophysiology and were able to show that these same cortical neurons grown in a Petri plate had a 50% decrease in the frequency of the AMPA receptor in both of the different lines that we have modeled. We've actually done other lines now too. So the structure is the same, but it's the excitatory potential that changes in patients that have deletions of this long non-coding RNA. So it's really critically important. It's the first time we've seen that characteristic feature in ASD. We don't know what the targets of this long non-coding RNA, this is what we're spending all of our time. But we think it's a new genetic entry into that big bubble of genes I showed you at kind of halfway through my talk. Now it's the first long non-coding RNA. So we need to see which protein coding genes it may be interacting with. We do know that it's actually probably going to the nucleus, perhaps binding chromatin. Very quickly, we've now done what I would call genomic surgery, CRISPR-based engineering of chromosomes. I was thinking about this when I was giving my talk. I'll come back to it later, but as this becomes more prominent in the clinical setting, I really think that chromosome-based surgery, and this is what we're doing here, perhaps it should fall into the Department of Surgery. It's something you can think about. But we can now use CRISPR-based technologies and delete exons so we can model this in the mouse. We've done this in many different ways. I won't go into the details, but it's pretty straightforward to do small sequence level changes using CRISPR engineering. We try to delete the whole 1 million base pair. That surgery doesn't work so well yet, but we're able to delete about 78,000 nucleotides of exon 4, 5, 6, and 7. With Paul Franklin's group, they're experts in neurobehavioral analysis. I won't go through all of this, but they can use different phenotype assays in mice. And Paul's student, San Yoon, has done a beautiful analysis now where he can show in the deletion model. So the region of the gene that's affected most in human, we then model that in mouse. the characteristics in mouse, and this is done completely blinded, are essentially what we would expect to see and what we've seen in humans. So the social repetitive behavior is slightly altered, slightly increased repetitive grooming. But there's just a slight decrease in muscle strength. They have all the kind of features you'd expect to see in mice that you would see in human of a kind of a high functioning autism. Again, the first time we've seen this in a mouse model, one of these autism genes. So we're trying to use these resources now to find out what the genetic pathway is and how it links to proteins that perhaps we can modulate using therapeutics. So it's a new entry point into autism. So I'm going to wrap up. I got just a couple more slides to get you thinking. If I can have, I know I started late, so give me a few more minutes. I think this will really get you thinking about from the surgery and the basic science perspective. You know, we're here where this is a slide I show a lot and I give public talks, stick out your tongue and say, ah, and we're now asking people to spit in a tube, right? So we can sequence a genome and we can actually do it in, the world record is 23 hours to go from spit into a genome through the sequencer, through the computer, back into a medical report. We can typically do it here in 4 days. And on, you know, if we work on a case, in a cancer case, for example, we can do it in about 2 days here at SickKids. So when people come in for their clinical visit, as we go forward, we're now into we do a whole genome sequence for about $750 US now on the research side. I think this is going to become standard to try to find out what the genetic form is going to complement all the other clinical data that goes into your input file to figure out, you know, what type of surgery you may need to do. So this is happening now already. In Toronto, there's over 20,000 different genetic counseling visits happening each year. And you know many of these people, but you can do sequencing at any stage of life. That's the really important thing, including non-invasive prenatal testing. You can do whole genome sequencing of fetal cells circulating in pregnant mothers. And this concept of genome engineering or surgery is happening in particular in somatic diseases all the time. It is so robust that it will become a form of surgery, I'm quite sure, going forward. Now, this is my last slide, Michael. This is an experience, this is unpublished. This is, I think, the conundrum here. We work with a cohort that was collected by Jen Crosby, Paul Arnold, and Russell Shakar here at SickKids, where they went to the Science Center in Toronto and collected DNA from kids. In this case, we did genotyping on 5,270. And they went through a series of computer-based testing and a very 15-minute survey. And they were categorized into different behavioral disorders like ADHD, OCD, ASD I talked about, and then other kind of learning difficulties, et cetera, shown on the y-axis here. And then some additional traits categorized based on the phenotypes. And then this was put into a continuous kind of Z-score type comparison. And then we did genotyping using a typical microarray that is very cheap. We wanted to do a cheap experiment. I think it was about $100 genotype here, a microarray. We'd like to do sequencing all of these. And then categorized all of the regions that were copy number variable found in different diseases we studied on my earlier slides, but never really found in controls. Okay, and the message here is that, you know, we find XXY whole chromosome CNV, and then we find a lot of these different deletions, 16p I talked about, single genes down here. And it's really the expression, the variable expression of the phenotype. So if you, for example, had a whole genome sequence and you found one of these deletions along the chromosome, you would know there's a higher risk of developing something like ASD or perhaps a learning trait, intellectual disability, but it's never going to be 100% predictive. Other than perhaps a few outlier cases like the PTCHDI-AS that I talked about, because we have more and more data accumulating. So the complexity is really how to make sense of all of the data. We're going to do whole genome sequencing and see if other genetic variants may play into this. And then both polygenic risk scores, et cetera, and of course what the environmental contributors are. But complexity is really the key. And then just to say 20% of the people in this study actually had a neurobehavioral diagnosis. Okay, these are typical kids coming to the Science Center. And then we found in 5% they had a rare CNV that likely explained at least part of that clinical presentation. So the numbers here are huge. And it'll take me to my last slide. This is typically the slide I show at the start of my talk. I thought I would move it at the end just to get you thinking throughout the day. You know, we do basic science really because the truth will be found in science. For the better part of probably 2 or 3 decades, the quote unquote blame for autism was on the maternal rearing of children, refrigerator mother syndrome, and then of course vaccinations. As I showed you, for at least 20% of the families we studied, we found the genetic factors involved now. And we can use that in earlier detection to lead to better health outcomes, in particular, also to be used in counseling. And then we've also found new therapeutic targets that we can start to begin to modulate, like the long non-coding RNA I talked about. And the approach really is to use individualized approaches based on the genetic alterations that are found. And as I said, for roughly 20% of ASD, we found key factors involved. What's happening in the other 80%? I think the Y chromosome is probably involved. It's combinations of genes, combinations of rare and common genetic variants. We need to get our numbers much more higher, much larger, so we can do proper statistical analysis. But most important from the societal perspective and from the perspective of medicine, we've now identified some of the genes that are involved in the features, as I said earlier, that make us most human, so-called decoding ourselves. Genes involved in cognition, speech and language, and social behavior. And again, 75% of the genes in the genome are involved in expressing the brain. So I think this is really the future. The last, I would say the last 20 or 30 years have been focusing on that 10% of genes that were enzymes, and then a lot of the genes involved in cancer, because that's where the funding has been. And now the future will be to kind of decode this last realm of genes, those involved in brain and human development. So with that, I'll end, and I'm happy to take questions and discussion if there's still time. Thank you so much, and great to see you again, Ben. I'm very honored to be invited to give this talk.
That's great, Stephen. Thank you. A wonderful, wonderful talk. Perhaps you could stop sharing your screen and we'll go to gallery view. We are a little tight on time, but I have a few questions that I've kind of been monitoring from the chat line and from the Q&A, and a couple of them are more clinically oriented and a few are perhaps a bit more research oriented. So one of the clinically oriented questions is that, so reports come back, clinical reports come back from the sequencing, and often the description is that there's a variant of unknown significance. And so how do you interpret this, and how will this evolve over time? Yeah.
So that's a 4-letter word. So it's really, okay, so and what happens is I showed you that figure with all the deletions and the bars, and that's kind of put into an Excel spreadsheet, and then many of you have seen these. And you'll see pathogenic variants, clinically relevant variants, and these things called variants of unknown significance. In a microarray or a sequencing report, these typically are variants that are found in disease populations, and then a much lesser frequency in control populations. So there's different databases, including the one that I showed you. There's a lot of studies ongoing. As the numbers increase, have more data to compare against, most of those variants of unknown significance move into the clinically relevant category over time. Christian Marshall and Jim Stavropoulos here at Sick Kids, and there's others in Toronto who have studied these. It's the area that my last slide touched on with the Science Center study. Okay, so most genetic counselors are learning how to deal with those. They keep their eye on those. And if you go back and look at the data a couple of years later, many of those variants of unknown significance then become clinically relevant. So the big issue is how often you go back and annotate the genome sequence, for example. That data is static. It's really good data. Maybe 10 years from now, we'll get better data using a new technology, but it's static. That'll be in the records. The key is to develop algorithms to go and relook at the data across the board and then recategorize. So there's whole industries developing around this, but it is a major challenge in the field. Well, I just give you a statistic. You know, we've done these studies and published a lot of this if anybody's interested. But so in a typical clinical genetic setting, you would get a result, cases coming in clinical genetics, about say 20% of the time you'll find a variant that's clinically describes the syndrome or relevance, 25, it depends on what it is. But, and then the variants of unknown significance you find in almost every other case. And depends on your resolution of analysis. If you go into sequencing, you'll find often 2 or 3 of these.
Okay. I won't have time to kind of go through all of the questions. Here's my apologies, but there was an interesting question from Neil Fleschner. And his question essentially is, you've been kind of talking about how these genetic variations negatively influence the phenotype. Can any of these mutations lead to athletic, musical, or intellectual advantages?
Yeah, so I should have mentioned this. So again, I took some of these slides out, but 1% of the autistic individuals are savants. They can have skills that are beyond the human realm, if you will. And many of the great mathematicians in history of science were probably on the spectrum. So I tried to get to this. I think this category of the genes in the brain, they're under very active selection, right? And the thing that's changed most in our evolutionary environment is actually our workplace and the need for processing of information. So the fine tuning at that so-called neurotypical neuron right now is under the greatest selection pressure. And it's actually happened in only 2 or 3 generations. So it's quite likely, and we've seen this now, there's that people are now meeting who are carrying alleles that lead to rare, say Asperger's type forms of ASD, that are meeting in the workplace at the Googles of the world, who are now getting together and passing those chromosomes on. This is the reason Google's interested in this project. 20% of their staff have Asperger's syndrome.
My goodness.
Okay, so yes, absolutely. These will tend to be the inherited variants involved, okay? Right. And it could be many combinations of these variants. That intelligence, IQ, cognition is a very complex trait. But ultimately, this is the kind of information you get. So you have to handle it very, very carefully when you're transmitting it back to the families.
Gotcha. Well, we are out of time, but I might ask either Jim or Ben or to make a comment or a question.
Well, I'm not going to ask. This has really been a fantastic talk, Steve, and it's been great to see sort of in my career how this has gone from just trying to understand the technologies for sequencing into something that's really been applied to clinical care. I think it's great, and I'm really looking forward to see things going forward. You're aware, there's things like the MedSeq project, which are randomized trials to see whether or not these make clinically significant differences. And I was going to ask you, but really kind of time about things like, we do phenylketonuria tests on all the newborns to screen them. It costs $17,000 for every phenylketonuria diagnosis. I think at some point this is going to become whole genome sequencing, $1,000 a pop. We'll do it for every newborn, keep them there, and it'll be the analysis of the data stuff you're talking about that's going to make the difference. So it's a great talk, and it's been amazing to see how much this has progressed during my scientific lifetime.
Yeah, I just add, this is really important. In the States, they have a project called All of Us. They're doing 1 million people. Okay, this is in the next couple of million genomes. The UK, they've already done over 100,000. They're doing millions. In China, they're doing tens of millions. So this data will be part of the clinical workup. And sick kids in many places in Toronto, it's happening. It's a little bit slower, unfortunately. It's a big data, AI, machine learning. This is where it's all at. It's dealing with the data.
The people who can interpret the data best are the ones who are going to be most ahead. And I actually think you're way ahead.
We do our best.
Jim, final comments from you?
Yeah, thanks, Steve. This has been a brilliant Ben Alman lecture. You've done Ben and the lectureship very proud, so thank you for that. I'm reading an interesting book now. It's called The Angel and the Assassin by Donna Nakazawa. I don't know if you've seen this book, but it's about the role of the microglia in the brain and how they impact on higher cognitive function and behavioral disorders in things like ASDs. So if you haven't seen it, I'll send it to you. We're way out of time for comments, but it actually adds another layer of complexity, like the non-coding RNAs and everything else you've mentioned. So it's a fantastic field, and we really thank you for giving this fantastic lectureship, Steve. So, and congratulations to you on all your work.
So I might then just bring things to conclusion. So Stephen, Professor Scherer, thank you very much for an outstanding lecture. It's just been, you know, wonderful to see the advances in science that have occurred. And I think that, you know, the work, I think, has a high degree of relevance for all of us practicing medicine. It'll be very interesting to see how this will be applied and how we might be able to predict some of the variations that occur in human response to disease, and how we might be able to modify the treatment. So just want to thank you. Thank you very much. Traditionally, you know, we have a few gifts and so on, and we'll get that sorted out offline.
They're in the mail.
Well, thank you very much. Okay, bye everyone.
Thanks, Steve. Thanks, Ben. Good to see you guys.
Thank you. Bye-bye. Bye-bye.